Good morning, students. Let us start with the new chapter, chapter number 6, Anatomy of Flowering Plants. What do you mean by anatomy? So, anatomy is the study of the plant parts at their tissue level. So, let us study how the plant parts are made and of which tissues they are made. So, following are the topics which we are going to study in this chapter. First, 6.1, the tissues. 6.2, the tissue system. 6.3, anatomy of dicot and monocot plants, root, stem, leaf, all three parts. 6.4, secondary growth in the flowering plants. So let us move ahead with the topic number 1, 6.1, the tissues. We can very easily see the structure, similarities and variation in the external morphology of larger living organism, both plants and animals. They are very evident. No need to go at the tissue level to find out the variations and structural similarities. Similarly, if we are going to study the internal structure, we also find several similarities as well as differences. This chapter introduces you to the internal structure and functional organization of higher plants. Study of internal structure of plant is called anatomy. Please underline the definition. Plants have cells at the basic unit. Cells are organized into tissues, and in turn the tissues are organized into organs. Different organs in the plant show differences in their internal structure. Within angiosperms, the monocot and dicot are seen to be anatomically different. Internal structures also show adaptations towards the diverse environments. Let us start with the tissues. A tissue is a group of cells. Please underline it. A tissue is a group of cells having a common origin. This is very important, and usually performing a common function. A plant is made up of different kinds of tissues. Tissues are classified into two groups, meristematic and permanent, based on whether the cells being formed are capable of dividing or not. So, meristematic tissues. Growth in the plant is largely restricted to specialized region of active cell division called meristem. So, wherever in whatever part, if the cells are actively dividing, that particular tissue is known as meristematic tissue. In plants, different kinds of meristems are found. The meristem which occur at the tip of root and shoot produce primary tissues known as apical meristems. Now we can see here in this figure apical meristem of root which is present just above the root cap. So that tissues are known as apical meristems. Here the shoot tip also consists of shoot apical meristematic zone in which the actively dividing cells are present. The minute dotted structure is known as root apical meristem and shoot apical meristem. Root apical meristem occupies the tip of the root, while shoot apical meristem occupies the distant most region of the stem axis. During the formation of leaves and elongation of stem, some cells left behind from shoot apical meristem constitute the axillary bud. Such buds are present in the axis of leaves and are capable of forming a branch of flower. The meristem which occurs between the mature tissues is known as intercalary meristem. They occur in grass and regenerate parts removed by the grazing herbivores. Both the apical meristem and intercalary meristem are primary meristem because they appear early in life of a plant and contribute to the formation of primary plant body. So these are primary meristems, apical and intercalary. The meristem that occurs in the mature region of root and shoot of many plants, particularly those that produce woody axis and appear later than the primary meristem, is called secondary or lateral meristem. They are cylindrical meristems, fascicular, vascular cambium, interfascicular cambium and cork cambium. So there are three types of secondary or lateral meristem: fascicular, vascular, intervascular and cork cambium. These are the examples of lateral meristem. These are responsible for producing the secondary tissues. So in the meristematic tissue, we have two types of meristematic tissues. First, primary, second, secondary or lateral. In the primary, we have two types of meristems, apical and intercalary. In apical also we have two types, shoot apical and root apical, while in secondary meristem we have three types. Following division of cells in both primary as well as secondary meristem, the newly formed cells become structurally and functionally specialized and lose the ability to divide. Such cells are termed as permanent or mature cells and constitute the permanent tissues. During the formation of primary plant body, specific region of apical meristem produce dermal tissue, ground tissue and vascular tissue. So the second type of tissues are permanent tissues. The cells of the permanent tissue do not generally divide further. Permanent tissues having all cells similar in structure and function, hence are called simple tissues. But sometimes permanent tissue have different types of cells and they are known as complex tissues. So permanent tissues will be of two types, simple and complex. Simple tissues. A simple tissue is made of only one type of cell. The various simple tissues in plants are parenchyma, collenchyma, sclerenchyma. First of all, parenchyma forms the major component within the organ. The cells of the parenchyma are generally isodiametric, means of similar size. They may be spherical, oval, round, polygonal or elongated in shape. Their walls are thin and made up of cellulose. They may either be closely packed or have smaller intercellular spaces. The parenchyma performs various functions like photosynthesis, storage and secretion. Let us have a look on how the parenchyma are arranged with the intercellular spaces. These are almost all isodiametric cells with a very little intercellular spaces. They are thin-walled as evident in the figure. Next are the collenchyma, which occurs in the layers below the epidermis in most of the dicot plants. It is found either as a homogeneous layer or in patches. It consists of cells which are much thickened at the corners due to a deposition of cellulose, hemicellulose and pectin. Collenchymatous cells may be oval, spherical or polygonal and often contain chloroplast. These cells assimilate food when they contain chloroplast. Intercellular spaces are absent. They provide mechanical support to the growing parts of the plant, such as young stem and petiole of the leaf. So basically they are meant for the support, mechanical support, because they do not have any intercellular spaces. So they are stiff in nature. They are hard in nature. So they are good for providing the mechanical support to the plant. Let us see how the cells look. Because their walls are thickened and they have depositions at the corners of pectin, cellulose and hemicellulose. So the cells are not having any intercellular space. They have a large vacuole and due to thickened corners and thickened cell wall, they are good for providing mechanical support. So these are the collenchyma cells. Now the next is sclerenchymatous cells. They consist of long, narrow cells with thick and lignified cell walls. See here, lignin deposition on the cell wall is found. So they have a very few or numerous pits, pore-like structures in their cell wall for the exchange of the things. They are usually dead and without protoplast. On the basis of variation in form, structure, origin and development, sclerenchyma may either be fibers or sclerids. Here we can see pits in the cell wall, very thin lumen, thick cell wall. Here the fibers are thick-walled, elongated and pointed cells, generally occurring in groups in various parts of the plant. The sclerids are spherical, oval or cylindrical, highly thickened dead cells with very narrow cavities called lumen. These are commonly found in the fruit walls of nut, pulp of fruits like guava. गुआवा खाते टाइम जो करकर जैसा लगता है बीच में स्टोनी हार्ड, दैट इज ऑफ स्क्लेरिड्स, pear and sapota, seed coats of legumes and leaves of tea. Sclerenchyma provides mechanical support to the organs. This was all about simple permanent tissues. We'll study about complex permanent tissue in the next lecture.